Okay, so I'm going to talk about the Penny Genetics assignment. Um, you guys will have paper copies of these available as well, but they're also on Google Classroom. I would personally prefer to do on paper because we're going to be like keeping tallies and doing some calculations. But either way is fine on my end. So you will have um, pennies that have been taped. So that one side, the head side, is marked with a capital T, the dominant allele, and the other side is marked with a lowercase t, which represents the recessive allele. In this lab, or I guess actually in reality, the big toes can be either short, which is dominant, a short big toe, meaning it's shorter than the toe next to it, or that's the dominant allele, and then a long big toe is a recessive allele. So the first thing you need to do is fill in on your paper. It says genotype / phenotype, and then it has the letters, the genotypes. So if it's two capital letters, that represents two short alleles, and so the phenotype should be short big toe is what you would write in that blank. Okay, so if I was doing this, I'm going to make myself a copy so I can edit it without changing everyone's. So if I was doing this, I would write short big toe. If it was a capital T lowercase t, that would also make a short big toe, just like the picture shows here. And then if it's two lower cases, right, that means that the person received the recessive allele from both parents, recessive, or both long alleles. So that's going to be a long big toe. All right. In this lab, we will make predictions using Punnett squares, and then we're going to use pennies to simulate the crosses. We'll compare our Punnett square results with our actual results. Just like how I've talked about before, the concept that just because something might be unlikely for two brown-eyed people to have a blue-eyed child, it's not impossible, and maybe they even have two children that have blue eyes. That doesn't disprove anything, so it's just that sometimes, the prediction doesn't match exact results. It's also similar to like the weather, right? We might say, we're expecting 4 inches of snow, and then we get 6 inches of snow. That's just, that's the actual result. The prediction was 4. It was fairly close. Okay, so part 1. Use a Punnett square to predict the ratio of offspring in a cross where the parents are both big T, little t. The Punnett square is set up right here. I can't edit this, unfortunately, but it would be big T in the first box, big T, little t in the second box. In the bottom left, it would be big T, little t. And in the bottom right, it would be little t, little t. You can pause this video if I'm going too fast. What proportion of the offspring out of 4 will be short toe, and what proportion would be long toe? Well, short toe, these 3 boxes would make short toes because they have capital T's in them. So that's going to be 3. And then only one of the boxes, little t in the bottom right, is going to make long toe. So this should be 3 and 1. Now you will determine the actual ratios by using pennies to represent crosses. You have two pennies. One side of the penny is the letter capital T, and the other side is the lowercase t. This penny represents a parent that has the genotype big T, little t. The second penny represents the other parent. One person is going to play the role of female, and the other will play the role of the male. When the coin is flipped, you're determining what sperm and what egg cell is being donated to the zygote, right? So, you can practice this. Make sure you have it down. So if you flip your 2 coins, did you get 2 capitals, capital and lowercase, or 2 lowercases? Write it here. So if I did this, I just did it and I got a big T, little t. So I would write big T, little t. But you write whatever you got right in that blank. It says, what is the phenotype of your offspring? Tall, or I should say long or short here. I would write short because mine has a capital letter and that's dominant. So mine would end up looking short. Okay, so just to make sure we understand, that's a little concept check before you get to the next part. Once we do that and we do it well and we understand, we're going to move on to the procedure. To determine actual ratios, we're going to flip 100 coins. If you don't flip 100 coins, you're going to have to do some math to modify your numbers. So I would recommend you flip 100, but if you get stuck, we can kind of address those changes when I'm back, or you can reach out to me for some help if I'm available. We're going to use tallies to record our data. Now, one thing I've seen with tallies is that students will just make lines and not do the 4 lines and then the slash for the 5th one. Please do 4 lines and the slash on your 5th one because it will help you keep track of your data without it being so hard to count everything up every time, because then you can count by 5s. Okay, so if we flip our coins again, you're probably going to flip the coin, your partner flips the other coin. On this time, I got 2 lowercase t's, so I would put a tally on this line down here. I'm going to use capital I for my tallies. Okay, and I just keep doing that. Okay, this time I got, oh wait, I flipped the other one. Oops, big T, big T. So I would put a tally up here. Oops. And I would keep going. That was 2 little t's. Okay, so you get the idea. Just keep making your tallies. Then once you get to 100, you're gonna write the totals over here. These will represent the percentage that it happens because it's out of 100. So if you got whatever you got, you're gonna add up this area. I'm gonna say, let's say I got 32. And we'll say I got 60 here. So I have 18 down here. Okay, that adds up to, oh no wait, that's too big. So let's actually make this top one 22. Okay, let's say those were my numbers. Okay, I know that part will take you some time. Please do it because you're gonna get better data if you actually do it. I'm just making up these numbers off the top of my head, and mine won't probably come out that well. Short TOE, so it says add big T with big T, little t. So I'm gonna add 22 to 60, so that would be 82. And remember, you can pause this video. For long TOE, it's just 18, right? It's just the only genotype that makes long TOE was little t, so I just copy that into here. And remember, these actually represent percentages because it's out of 100. Okay, it's right here. It says, since you flipped 100 times, your totals above represent a percentage. Your proportions from the Punnett square in your prediction also represent a percentage, where 1/4 will equal 25%, 2 out of the 4 boxes would be 50%, 3 out of the 4th would be 75%, and 4 out of 4 would be 100. So we're going to compare our predicted results to our predictions to our actual results. For big T, if I go back to my Punnett square on because this says predictions, from Punnett square from part 1, go up there. This box, my top left, was the only one that was big T. So that is 1 out of 4. So that should say 25%. Big T, little t. I had this box, the top right and the bottom left were big T, little t. So I'm going to go down here. 2 out of 4 would be 50%. And little t was the bottom right. So that's 1/4. So that would be 25%. Obviously my numbers here should add up to 100 because it's the genotype totals. Okay, 25 plus 50 plus 25 is 100. So I did well there. My predicted for short versus long TOE, remember I said 3 out of the 4 were short. Over here we wrote 3. 3 out of 4 is 75. So I'm gonna write 75%. And 1/4 is still 25%. You could also get this 75% by just adding big T to big T, little t, right? That would make 75%. So, all right, now our actual flips. This is where it gets kind of interesting. You're gonna copy your information for these top 3 rows that you did over here. So mine should say 22, 60, and 18. 22%, 60%, and 18%. Oops. For short TOE percentage, that's over here where we added our percentages together for what genotypes would make short TOE phenotypes. We got 82%. And then it was still 18% for long TOE phenotypes. Okay, so now we can side by side compare our predictions to our actual. And you notice that they're not exact matches. Mine are actually pretty far off. Hopefully yours are closer than this. Would you consider the predicted values to be the same, close to the same, or not at all the same? I'm going to say close to the same because, I mean, they're not super far off, but you can see my heterozygote number is pretty far off. It's pretty high. There's a big difference between a 60 and a 50, but still, it's not wildly different. So I'm going to say close to the same. Now, on part 2, we're going to repeat the procedure, but this time we only have one heterozygote parent. The other is homozygous recessive. We do not need to flip a coin for this parent because we know they're always giving a lowercase t. For homozygous parents, we don't need to flip. We only need to flip for the heterozygote because this has a 50-50 chance of giving an allele, so we don't really know what it's going to give. But we know 100% of the time this parent is giving a lowercase t. So you can just leave, you can just take one coin and flip it. But before we do that, we have to set up our predictions. So if I set up a Punnett square for this, and you're supposed to do this on your own, write a little window, put one parent across the top, one parent across the side. And so you're going to get 2 squares that are big T, little t, and 2 squares that are little t, little t. So that's, we predict 50% to be short toe and 50% to be long toe. And you would see that if you fill in your Punnett square. You do need to show me your Punnett square on here. Don't just copy this. You should also have a Punnett square that proves it. This has replaced one of your pennies with a chip that's a little t, but honestly, we don't need to do that. We can just flip one of them, if you feel better about just leaving one of your pennies on the lowercase t face up and just don't touch it, that's fine too. But we really only need to flip one. So if I flip 1, I got a capital T. I would put a tally over here because I know one parent is giving that lowercase t, and it's just this heterozygote parent that I'm flipping for. Now I flip that same coin again, and I got another capital T, so I'd put a tally here. And I would keep doing this again until we get to 100. This time it's a lowercase t, so we know the heterozygote's giving a lowercase t, and that other parent is always giving a lowercase t, so that's going to be 2 lowercase t's. Flip a coin again, lowercase t. Okay, so you're going to keep doing that until you get to 100. And again, that way these will represent percentages. What percent of your offspring are short toe? I'm going to do a couple more just so I have some numbers to go off of. All right, so let's say I got lowercase t. So I'm going to say I got, let's say I got 60%. I'm going to say I got 60 that are this, and 40 that were heterozygotes. So if I did 100, let's pretend that's what I got. What percentage of your offspring were big T, little t? That would be 40% because out of 100. What percentage are short toes? Well, that's the genotype that makes short toes. This genotype makes long toes. So again, 40%. I'm going to delete. There we go. What percentage are little t? 60 out of 100, so that's 60%. And what percentage are long toes? That's going to be 60% because it's the only genotype that makes long toes. Now compare the predicted ratios to the actual ratios. So we predicted a 50-50. So those should be 50% there. Actual from flips, I'm saying I got 40% for short toe and 60% for long toes. And that's it. That's part 2. It's done. Now we have some analysis questions. I will go over these answers with you when I am back, but you should attempt them. They should not be left blank. And then we'll go over the answers when I'm back. You'll have an opportunity to correct your work, but if you leave anything blank, you'll be docked some points. Please make an attempt. You can try to research some help or talk to your partners, but I don't expect you to get these all right on your own. I just expect you to try and fill them in. Okay, so we kind of talked about some of these already, right? Like why are predicted ratios rarely the same as actual ratios, right? Think about, the weather. Just because we say we're expecting it's a 10% chance it's going to rain, that doesn't guarantee we're going to have rain of a certain volume. Or same with snow, right? We expect 4 inches. That doesn't mean we're getting exactly 4 inches. Okay. All right. You're going to do a Punnett square on this one. All right. Good luck. And I will see you guys very soon on, I think it's Thursday.